Our next outstanding presenter is Professor Chong Liu from Hangzhou, China. His abstract is entitled Olfactory Sensory Experience Regulates Gliomagenesis via Neuronal IGF1 Signaling.
Thanks, Sean, for the introduction, and also thank the organizers for this opportunity to share our findings on this webinar. My name is Chong Liu from Zhejiang University, China. The research interest in my laboratory is to understand the molecular and the cellular mechanism of gliomagenesis. Although most groups in the field are focusing on the topics at the late stage of tumors, such as tumor heterogeneity and cancer stem cells, etc., we are more interested in knowing how normal cells transform into malignancy. That is also called the tumor origin issue and how early progression happens. Today, I would like to share some of our findings about how neurons and neuronal circuits function as tumor microenvironment for glioma initiation and early progression. Actually, my interest about this question can go back to 12 years ago when I did my postdoc training. In our previous work, we developed a genetic mosaic system called MADAM to model gliomagenesis and identified OPCs as a soft version for glioma. Interestingly, we noted at the time that throughout the entire progress of tumorigenesis, OPC-like tumor cells always interact with neurons. When co-culturing tumor OPCs with neurons together, tumor OPCs can make complicated connections with neurons. These hints strongly suggest that neurons might play important roles in tumorigenesis. Indeed, in the past 8 to 10 years, many significant findings have been made to support this possibility and clearly show that neurons and tumor cells can influence with each other in the brain. These findings together come to the blooming of a new field called cancer neuroscience. On the other hand, external stimulation has also been proved to affect the growth of grafted tumors in vivo. Dropping the tumor grafted new mice into an enriched environment can suppress the growth of grafted tumors. It is generally believed that enriched environment regulates tumor growth mainly through indirect effects contributed by the immune and endocrine system. However, thinking about the unique microenvironment of brain tumors, where tumor cells can directly interact with neurons, the possible that the external stimulation can directly affect the glioma behavior rather than controlled by something outside of the brain. Therefore, we made a very simple hypothesis. That is, what variant external sensory stimulations or even thinking, cognition, and emotional changes can directly affect the gliomagenesis through the corresponding neuronal circuits. To test this hypothesis, we took advantage of a genetic mouse model that can recapitulate gliomagenesis originated from adult OPCs. As the mutant cells can be labeled with red fluorescence protein, when the initial driver mutation is also introduced, the entire process of gliomagenesis can be clearly visualized and traced. When we characterize the tumor incidence and the locations appeared in this tumor model, we found that tumor frequently localized in the anatomical structures related to process olfactory information, and the olfactory bulb is the place where tumor first appeared. Take a closer look. We found that tumor initially appeared in the GL layer, which is the first relay in the olfactory circuit to connect the olfactory receptor neurons in the olfactory epithelium in the nose cavity with the mitral or tufted cells in the olfactory bulb. Interestingly, tumor frequently appear in the lateral and ventral side of the GL layer, where are also the place with daily active olfactory input. Based on these observations, we asked whether olfactory inputs can directly regulate gliomagenesis, at least in this model. To test this hypothesis, we insert a small plug into the nose cavity to deprive olfactory input. We found that unilateral olfactory deprivation can significantly suppress the gliomagenesis on the same side of the olfactory bulb. In combining with other genetic evidence, our data clearly show that olfactory experience can directly regulate gliomagenesis. To further determine the molecular mechanism, how olfaction controls gliomagenesis, we did bulk RNA sequencing for nose cavity blocked olfactory bulb tissues and found that only 17 genes were downregulated after olfaction deprivation. Among them, IGF1 drew our great attention, not only because IGF1 is the only growth factor shown here that significantly promotes tumor growth in culture, but also because its expression is neuronal activity dependent and our function are regulated. Nicely consistent with this observation, in our previous study, we have proved that IGF1 signaling pathway is essential for OPC malignant transformation in the same genetic mouse model. All of these lines of evidence together strongly implicate that olfaction may affect the gliomagenesis through IGF1. Indeed, we found that IGF1 is highly expressed in the olfactory bulb, nicely overlapped with the tumor hotspot in our model. By using multiple cellular markers, we identified that mitral tufted cells are the major cell types in the olfactory bulb to strongly express IGF1. We next specifically knock out IGF1 in mitral tufted cells and clearly proved that knockout of IGF1 in mitral tufted cells can suppress tumorigenesis in the olfactory bulb. Therefore, proved that mitral tufted cells are the major cellular source of IGF1 in supporting OPC transformation. These findings also raise the possibility that neuronal IGF1 might be a potential therapeutic target for glioma treatment. To explore the human relevance of our mouse model-based studies, we performed functional MRI imaging experiments and confirmed that the insular cortex is one of the most activated brain structures upon olfaction stimulation in humans, along with the piriform cortex. We also analyzed a pool of adult patients with oligodendrogliomas, which are putatively believed to be originated from OPCs. After normalizing tumor frequency by the volume of associated brain structures, to our surprise, we found that the normalized incidence of oligodendrogliomas were much higher in the insular cortex than any other anatomical structures. This correlative data collectively provide compelling evidence supporting our mouse studies and suggest the further studies of the causal link in humans between olfaction and the glioma. To summarize today's talk, our work strongly suggests that the functional neuronal circuit can work as a hub to connect the external microenvironment and the local microenvironment, and play important roles in the initiation and early progression of gliomas. In our current study, we demonstrate that olfaction input can affect gliomagenesis in the olfactory bulb through IGF1. Independently, the Gutman and Monge's lab proved that visual stimulation can affect the optical nerve gliomas through a completely different growth factor analogenesis rate. These findings together raised an exciting possibility that different types of gliomas may co-opt with distinct neuronal circuits for their better fitness during tumor initiation and early progression. I would like to thank my team and many of our collaborators. This work was mainly done by a talented graduate student, Pengxiang Chen. And I will stop here, and I'm happy to take any questions.